Okay, good morning everyone. Thank you very much for giving me this opportunity to present some of my work. As the chair introduced, I'm trying to give a spin on the title. Just my emphasis is on ROS, that is reactive oxygen species molecules, which are highly notorious, especially in the case of diabetes and its complications. ROS, you all know the basics, I hope. It is chemically reactive oxygen molecules, molecular oxygen. There are different types of ROS molecules, from superoxides to peroxide, hydrogen peroxide, hydroxyl ions, hypochlorite ion. Lots of ROS molecules are present, and it is very fashionable to work in this area because it is shown that ROS has multiplicity of roles, from cancer to diabetes to inflammation to whatnot, most of the diseases. As you all know, this ROS is produced primarily in the immune system. So the body has a mechanism to produce these ROS molecules. That means there should be some functions for these ROS molecules. The primary function is to fight the infection. So whenever we get some bacterial or viral infection, these ROS molecules are produced inside the phagocytes, and they attack the external organisms, microorganisms, and to kill them. However, unfortunately, sometimes it will start attacking our own cells. Rather, there are some physiological functions. It has been shown that ROS molecules are important signaling molecules as well in fetal development in womb, or in thyroid secretion. Various kinases, enzymes are activated or inactivated by ROS signaling. Inherently, we have developed antioxidant mechanisms. So ROS molecules are produced in our cells, and once their function is over, they should be quenched by antioxidant mechanisms. You might be aware of various vitamins like vitamin C, selenium, different micronutrients are involved in this antioxidant metabolism. Apart from that, we have different enzymes like superoxide dismutase, catalase, different enzymes to take care of these ROS molecules. So there should be a balance, how much ROS is produced and how quickly they are quenched or degraded, to maintain a healthy balance in every cell. As I told, ROS pathology, that is ROS mediated pathologies, are widespread, starting from inflammation to liver to alcoholic disease to neurodegenerative diseases. If the ROS is excessive and our antioxidant mechanism is ineffective, as I told, ROS will start attacking our own cells, DNAs, proteins, lipids, different molecules, and cause cell death and cell damage, which can lead to any kind of disease, as I mentioned. However, early trials with supplementing with like wholesome antioxidants were not very encouraging. Like the results were not very positive. That could be because we need these ROS molecules to do various physiological functions. They are not totally pathological. So there should be a targeted approach in this case. Okay, diabetes, this metabolic syndrome, this I need not introduce to this august audience. Diabetes is no more a single disease. It usually is present in obese people with hypertension, some cancers, liver diseases, heart diseases, etc. So we have to look the picture in wholesome, and it is widespread throughout all continents. Sad part is around 50% is undiagnosed, especially this part of the world where I come from, Southeast Asia and South Asia. Almost 1 in 2 people who are diabetic are not diagnosed. So health awareness is another important aspect to work on. This is a growing problem as per WHO statistics. It is going to increase by 55% by 2035. So it's an immediate need to address. But rather than directly on diabetes, I work mainly on the complications. As you all know, diabetes can affect any organ of the body. Since it affects the blood vessels are everywhere. They are there for all the organs. So indirectly, diabetes can affect any organ because it affects the bigger vessels, macrovessels, or smaller vessels like capillaries and arterioles, microvascular. So various diseases I have enumerated here. If you come to ROS, there are multiple pathways by which increased glucose or hyperglycemia induces ROS production. One of the two main pathways I have listed here, one is via glycation reaction, that is glycation or glucose addition of the proteins leading to advanced glycation end products. As a byproduct, ROS is produced. Another is a traditional TCA cycle. People who have done biochemistry will know this electron transport chain or respiratory mechanism through which ROS is produced. So, although my work is mainly on vascular disease, to do justice for my topic, I have to start from the stillbirth, that is before being born. Stillbirth is the dead born, the baby is given birth as dead baby. One of the main reasons is gestational diabetes. One of the main causative agent for gestational diabetes induced stillbirth is excessive production of reactive oxygen species by the placental cells like trophoblasts. So, we are trying to find out what could be the mechanisms behind ROS production, ROS excessive production in gestational diabetes in trophoblast. I am not going to give the data on that, I am just touching upon these. Second aspect is diabetes induced ROS production in macrophages. Normally, as I told, macrophages produce loads of ROS to take care of the external attacks like immune function, host defense, but excessive ROS in macrophage can be an important contributing factor for production of foam cells in atherosclerosis, especially in diabetes. So, we are looking at the mechanisms behind ROS production in atherosclerotic foam cells as well. Third thing is neuronal ROS in diabetes. As you all know, brain consumes maximum amount of oxygen which is going inside our body, which we are taking into our system. So, obviously, the higher concentration levels in brain could be a reason behind the excessive production of ROS there, and also for synaptic communication or transport, some ROS molecules are used. But unfortunately, excessive ROS can lead to neurodegeneration, various neurodegenerative diseases, sensory neural deafness, some diseases like multiple sclerosis or Alzheimer's. The ROS is implicated. So, we are trying to look at these as well in terms of memory and learning defects, diabetes affects that. So, ROS could be a mechanism behind that. Okay, coming to the results I am going to present, I am going to give some idea about ROS production rather than defective antioxidant mechanisms in vascular system. In case of diabetes, as I told, there are various mechanisms through which ROS is produced in diabetes. I am going to especially concentrate on endothelial cells, because endothelial cells, as you all know, produce this wonderful molecule called nitric oxide, which is a magical molecule, and it has been awarded rightly Nobel Prize for the person who discovered it. It causes vascular relaxation, which is absolutely essential, especially in people with atherosclerosis and diabetes, in which obviously blood supply is compromised. This nitric oxide is, as I told, has multiple functions, almost always beneficiary. It is antiplatelet, anti-inflammatory, anti-proliferative, antioxidant and vasodilator. But the question is, our group initially discovered that total nitric oxide availability, or rather bioavailability in endothelial cells in case of insulin resistance and diabetes mellitus type 2 is reduced. And we have also found an important ROS molecule called superoxide, which is a quencher for nitric oxide, is produced excessively in endothelial cells of insulin resistant mice and patients. So, we wanted to look further deep into it. What could be the source of this excessive superoxide in endothelial cells, and we have zeroed in on an enzyme, that is NADPH oxidase, or NOx in short, which is the culprit enzyme for the production of superoxide in various cells, especially in endothelial cells. An isoform called NOx2 or NADPH oxidase 2 is the most important source of superoxide molecule. So, we tried to look at it. We have used different strategies, pharmacological, genetic, to inhibit this NOX2, and also we have developed some knockout mice to delete this NOX2 and C. The pharmacological agent we have used is a peptide, that is GP91DS STAT peptide, which is, in short, is an inhibitor of NOX enzymes. We have directly measured superoxide, and we have also measured the vascular function in these mice. So this is nothing but vascular relaxation curve, that is in response to acetylcholine, the iota of mice, how quickly they have relaxed. So better the relaxation or quicker the relaxation, it is better. We have used two models of insulin resistance, that is endothelial specific insulin resistant mice, genetically manipulated, and also whole body insulin resistant mice, again genetic transgenic mice. In both of these, the vascular relaxation was impaired, but when we treated these vessels, that is aorta, with this peptide which inhibits NOX, it was significantly improved to that wild type level. So it basically shows that we can pharmacologically inhibit the NOX enzyme so that we can improve the vascular relaxation. We have directly measured superoxide as well, different assays, but basically the results is same. The superoxide production was higher in insulin resistant mice endothelial cells, and NOX enzyme inhibitors could reduce it significantly. To reiterate the point, we have used genetic method, as I told, that is knockdown of this NOX2 mRNA using siRNA. In this endothelial cells, when we transfected with siRNA against NOX2, we could significantly reduce the superoxide production as shown here in the down figure. Another thing, rather than directly putting the treating the vessel with the peptide, which is NOX inhibitor, we have implanted a small drug delivering device into the mice and delivered this drug over 28 days period, then measure the vascular function. In both of these, our insulin resistant models, infusion of this peptide, that is inhibitor of NOX enzyme, could significantly improve the vascular function, that is vascular relaxation. To nail the point to nail the point, what we have done is we have totally knocked down the NOX2 gene in the transgenic mice, that is insulin resistant transgenic mice, then measure the superoxide level in the endothelial cells and also measure the vascular function. As the first two figures show, the superoxide production was significantly reduced when we knocked down the NOX2 enzyme totally, and the vascular function was significantly improved. So what we have shown is this NOX2 enzyme is a culprit in case of endothelial dysfunction in patients with type 2 diabetes. So by targeted therapy against this NOX2, we can improve the vascular function. If you remember what I told at the initial part of this talk, that the large scale trials which have in total tried to reduce the ROS production by introducing antioxidants, non-specific antioxidants, have failed. So we believe that one of the reasons, as I told, could be the side effects because of normal physiological functions of ROS also inhibited, were also inhibited in those trials. But we have shown that by specific knockdown or specific inhibition of particular source of NOX in particular types of cells, we can still improve the functions, especially improve the complications or treat the complications of diabetes. So we should have a local or controlled delivery, and we have to identify specifically what is the source of this specific ROS, which is the culprit in our disease of interest, and we have to target that rather than wholesome targeting. Okay, that's what I want to say. I just want to thank all our collaborators and the students who have done the work. Finally, thank you for listening.
Thank you very much. Are there any questions about this topic? So you had the evidence from the animal models, but how does it work in the humans?
In the humans, we are in the process of studying, but we have just proved the mechanism, but the treatment aspect we cannot, because this peptide is not approved for human trials yet.
Okay. Okay. So thank you very much. Here you have your.
Thank you.
Thank
Thank you.
you.